Καλησπέρα σε όλες και όλους. Ονομάζομαι Αλέξανδρος Τζαχρίστας, είμαι εινολόγος, μέλος του Δούσου της Πανέπο και σήμερα θα είμαι ο παρουσιαστής και ο συντονιστής της συζήτησης. Πρώτα απ' όλα θα θέλαμε να σας ευχηθούμε καλή χρονιά. Είναι η πρώτη παρουσίαση για τη φετινή χρονιά με ένα θέμα έτσι πάρα πολύ ενδιαφέρον. Αρχικά να ενημερώσουμε ότι όλη η προβολή θα μαγνητοσκοπηθεί έτσι ώστε να είναι διαθέσιμη στο κανάλι της Πανέπο. Σας προτρέπουμε να εγγραφείτε έτσι ώστε να έχετε πρόσβαση τόσο σε αυτή όσο και στις προηγούμενες παρουσιάσεις ανά πάσα ώρα. Το σημερινό θέμα έχει να κάνει με τους μικροοργανισμούς αλλίωσης. Γνωρίζουμε ότι το κρασί είναι ένα κατά γενική ομολογία ασφαλές τρόφιμο, όμως αναφορικά με την ευαισθησία του στους μικροοργανισμούς αλλίωσης έχει παρατηρηθεί ότι σε αρκετές περιπτώσεις μπορούν να παρουσιαστούν εκτροπές. Έχουμε τη χαρά να έχουμε μαζί μας τον κύριο Παναγιώτη Θαμνίδη από την Ιννοανάλυση και την διδάκτορα κυρία Καμπολόγκο Σιμώνα, οι οποίοι θα μας μιλήσουν για το κρίσιμο θέμα αυτό. Okay, Simona, I did a small introduction. I let Panayotis speak briefly about the company and what he's up to, and then he will give the floor to you. Okay? So, Panayotis, you can take the floor.
Okay. Σήμερα θα μιλήσουμε για κάτι το οποίο παρότι η εμπειρία μου τώρα έχει δείξει ότι σχεδόν πολύ λίγοι κάνουν έλεγχο. Πλέον στα σύγχρονα εινοποιεία και σε πολύ καλά εινολογικά εργαστήρια γίνονται η πλειονότητα των αναλύσεων με ασβέστια, χαλκούς. Πολύ λίγα εργαστήρια όμως και πολύ λίγα εινοποιεία κάνουν μικροβιολογικό έλεγχο. Και είναι κάτι το οποίο νομίζω λείπει. Η σημερινή παρουσία θα αφορά κάτι το οποίο θα λύσει τα χέρια πολλών εινοποιών. Αφορά λοιπόν την παροχή κάποιων διαγνωστικών τεστ, διαγνωστικών kit για την ανίχνευση spoilage μικροοργανισμών, bread, γαλακτικών βακτηρίων, οξικών βακτηρίων, εύκολων στη χρήση, χωρίς να χρειάζεται να επενδύσει σε μικροβιολογικό εξοπλισμό, να στήσει μικροβιολογικό εργαστήριο ή να στήσει PCR, εύκολων, γρήγορων, ώστε να μπορείς εύκολα, γρήγορα και οικονομικά να έχεις μια ανίχνευση πιθανών spoilage μικροοργανισμών μέσα στο εινοποιείο σου. Είτε κάποιο βαρέλι μολυσμένο με μπρετ, είτε κάποια γαλακτικά βακτήρια τα οποία να είναι τα λεγόμενα spoilage γαλακτικά βακτήρια και όχι τα καλά που κάνουν τη μιλογαλακτικά ενώ κοκκουσόνη. Άρα νομίζω θα είναι πάρα πολύ χρήσιμο να παρουσιάσουμε μια τέτοια δυνατότητα. Η εταιρεία, η κυρία Σιμώνα Καμπολόκο που θα μιλήσει, στην ουσία είναι η ιδιοκτήτρια CEO της εταιρείας και η εταιρεία κατά βάση η BioSelf ασχολείται με μοριακή ταυτοποίηση ζυμών και επιλογή ζυμών από μόνο ζυμομυκήτων μέχρι και παραγωγή σε πιλωτική κλίμακα, και ένα από τα κομμάτια που έχει φτιάξει είναι και την ανίχνευση αυτών των τεστ για ανίχνευση spoilage μικροοργανισμών στον οποίο. Θα δώσω το λόγο στην κυρία Σιμώνα που αυτή θα κάνει την παρουσίαση. Εγώ έκανα ένα πρόλογο. Señora, Doresa Simonia Capolongo, and now it's yourself. I make a small introduction about what we're going to say today, and you can take the phone and you can start your presentation. Present your company and everything.
Thank you, Panayotis. Thank you. I'm Simona Campolongo, and I am the owner of a company based in Italy, in Turin, that is called BioSelf. Our BioSelf is a small but innovative company, and we are specialized in yeast innovation. I will explain what I mean with yeast innovation, and also on fermentation technology for the wine industry, also for the beer, but especially for the wine industry. Our expertise is developing some tools like self-breath and self-wine for winemakers. And now I'm going to explain what they do and how they work to provide wineries with some solution to help them to analyze their wine without sending any sample in other laboratories. Our team is made from me, from the co-founder that is called Matteo, and our lab technician that is called Federica. And our mission is to actually we have patented these two products, self-breath and self-wine, but we continuously study and improve research and innovation idea to help the innovation for independent and small or medium winemakers. I mean the wineries that doesn't have a laboratory. Our mission is to transform the winemaking processing, giving the possibility to the enologists to have some tools for the autonomy in to do some autonomous analysis, and also to check constantly the quality of their wine and also of their grapes. We want to help winemaking to be more sustainable, avoiding wine spoilage. Our product, we have some product, but also we provide a very useful service. Our product is self-breath. They are all called self because the idea that we want to transmit is that it's something that you can do by yourself. You don't need to send the wine samples somewhere else. And you don't need any help from external resources. So our products are called self-breath for the analysis, especially only of the Brettanomyces yeast. Self-breath for grape to understand if the grapes have a Brettanomyces content before they arrive in the wineries. So it's something that is possible to do already in the vineyards. And also self-wine that can help enologists to understand the entire wine microbiology. I will explain later. And we have also a service called the Self-Yeast. This service, it's called Self-Yeast, but enologists cannot do this by themselves for all the steps, but they can do just the first step by themselves, sampling the must during fermentation and sending us the samples so we can extract some yeast and then study them. Where it's possible to use our product. Actually, in all the point of the winemaking process, from the harvesting, actually, even when the grapes are still on the vine, because with self-breath for grape, you just need to take some samples. But then you can use our kit during harvesting, before crushing, during the fermentation, during aging, filtration, and even before bottling, to understand before bottling if there are some microbiological problems. What I want to say is actually something that I don't know in Greece, but at least in Italy, some wineries have understood that they need to pay attention to prevention, but not all of them. Preventing breath or bacterial contamination is really important to maintain wine quality. Because the prevention starts to identifying the critical control points. So the wineries needs to minimize the risk of breath or even other spoilage microorganism from the introduction from external sources. They need to evaluate the sanitization efficiency. And actually, it's something that I didn't mention, but our products are suitable even to analyze the washing water. And so you can use also to understand if all the procedure of sanitization of winemaking equipment, storage tools, and wineries can be successful, and even to monitor the contamination risk during the critical stage, like aging. Let's talk about Brettanomyces. One of the biggest problem of of wine is Brettanomyces, and as you can see, is a yeast that can produce different smelling compounds. The most famous one are ethyl phenols, but can also produce vinyl phenols, volatile phenols like 4-ethyl phenol, 4-ethyl guaiacol, but also for vinyl, the vinyl phenols, and they also have a smell. But it can also produce acids like acetic acid and other acids that you can smell, obviously, like the normal acetic acid smell, but even like the similar, like an aged cheese. It can produce also some esters and lipid oxidation degradation product. Usually, the breath smells are wine bed odors like horse, barnyard, stable, wet dog, and usually the possibility of smell it is between 400 and 600 of micrograms per liter. After this, the consumer usually can smell it, and it's not happy to drink this wine. One of the typical misunderstanding of wineries is that Brettanomyces can only be in wood, but actually it's not true, and even the literature says this. Brettanomyces can live everywhere. At the moment, there are some rapid tests available, but they are very expensive because you need to. I will show you later, but you need to buy an instrument and also all the reagents. Instead, we have invented actually and patented those products. So there is the possibility to offer a rapid and economic solution to detect in an early stage Brettanomyces. How our products are made? They are actually ready to use. They have an agar slide. They arrive as the picture you can see. They have a lid, and in the lid there is an agar slide already ready. You don't need to do anything. You don't need to add the water. Anything is ready, and is inside a plastic container that you have to fill with some milliliters of wine. Here you cannot see the label, but in the product there is a label that sign the level until you have to fill the wine. How to use our product? You have to remove the cap. You place it out. Do not touch the agar. Then you pour the wine until the line that is signed. You close and shake gently for just 1 minute. Then you empty the container. You drain a little bit self-breath or even self-wine. You drain just a little bit with the tip of the slide on a paper. Then you close and leave it at room temperature and wait. Here we have written 10 days, but actually the bad smell arise already after 2-3 days. Is the first sign that you can understand, because actually our product they have this functionment. What it happens is that color will change from blue to yellow. Then you can also smell the breath odors, and you can observe if some yeast colonies will be appeared on the agar. If all these 3 points says yes, you have a contamination, and you can also understand the contamination level from poor, from a small contamination, medium contamination, or high contamination. Then you can decide what to do with your wine. You can filter, you can add chitosan. There are different things that you can do. Here I show typical results. There is a new one that is blue, a negative one that is more or less green, and a positive one where you can see that the color is completely changed from blue to yellow. And also there are some colonies. Obviously, from a webinar you cannot smell it, but I can assure you that if you have a contaminated wine, after a while you will smell the contamination. For the other things of other types of problems, of microbiological problems, we have studied and developed self-wine that is made of 2 different vials with the same technology, that is a deep slide technology. And what we detect is acetic acid bacteria, wild yeast, and lactic acid bacteria. For the wild yeast, we can also detect Brettanomyces bruxellensis. So you can say, but if you detect Brettanomyces bruxellensis in self-wine, why I should buy self-bread? Because it's much clearer and is also faster. It functions self-wine as a functionment similar to self-bread. And here there is a picture that can show you better the contamination level. So you can see there is a low contamination, a medium contamination, and a higher contamination. On the green, with the vial with the green lid, we detect yeast, and on the red lid we detect bacteria, even the Enococcus ceni. Which is our competitive advantage? Here I have studied our competitor, and as you can see, I don't know in Greece, but in Italy the microbiological plating from a laboratory range between 15 and 22 euros. But the results can be even delivered after 15 days. But there are other kind of analysis that you can conduct, but usually you need to invest a lot of money for the instrument, and even for a qualified person that can do the analysis for you. So our product actually are cost are affordable, are reliable. They have a long stability because they have 2 years of shelf life for self-bread and 1 year for self-wine, and they are very quick and easy to use. The last but not least things that I want to share with you is that we are the yourself company is young, but I've been working for another company for almost 10 years, and what we are able to do is and we are even very good to do this, yeast selection. Indigenous yeasts are some yeasts that naturally occurs for in specific vineyards on cellars, and they are grown on these vineyards, and they have how to share this, the ability to use the compound, the special compound produced from that variety. So they reflect the local environment and they contribute to terroir expression. I don't know if you have any experience of this, but when we do some small micro vinification, when the mast is the same for everyone, and the unique variable is the yeast, and there is a really big there are really big difference between the wines between them. So the yeast can enhance aromatic richness and the unique sensory profiles, and promotes natural fermentation with a minimal additives. Furthermore, you don't need to buy the yeast from the multinational. I don't know about the Greek market, but in Italy we have 2 or 3 companies that product, after our selection, can product the yeast in a creamy form like a yogurt. This selection works in this way. We do, or actually the enologist does a sampling, collect grape and must from the vineyards or fermentation tanks, and send to us. We do an isolation, so identifying yeast strains, and for this we use some molecular techniques like PCR. Then we do tests for fermentation performance, for aroma production, and compatibility with the goals of each wineries. The application is to have a wine that has a unique taste, because something that we do with our clients is that we send or we bring, if they are closer, the micro fermentation wine, and we choose with them the final wine, and usually they are really amazed from the results. They usually do not expect that the yeast can have this effect on the wine. So in conclusion, we are really innovative because we have launched on the market a disruptive and patented product that can help wine and also beer sector to improve quality and to decrease waste and wine spoilage. If you want to follow us, you can find us on Facebook or on Instagram, and also feel free to write me for any question. If you want to try the product or have specific question, you can refer to Panagiotis. This is our resellers distributor in the Greek area. For me is all. Thanks.
Okay, Simona, thank you very much for the detailed presentation. We have one question from Dr. Maroula Dimopoulou. She asks, can you please tell us the detection level of self-bred in terms of colony-forming units per ml?
Yes, it's 10 cells per milliliter. I can understand that is quite incredible, but probably we have imagined that the agar, the gelatin, act like a magnet. So when you add the wine, the 30 milliliters of wine, when you shake it, the agar attract the cells on this surface, and actually the detection level is for self-bred 10 cells per milliliter. Instead, for self wine, the level. Let me share it again in just a second. The level can change depending on the strain. So we have seen that for some of them, can you see them?
Yes, we can.
For some of them, the quantity level, the minimum quantity level, is always 10 cells per milliliter, and for others, it's 100.
Okay, so thank you very much.
You're welcome.
I don't see another question. I would like to ask, what's the principle of the method?
It's just a culture-dependent method. There is a selective media on the slide. We have developed. I did a PhD on Brettanomyces, so actually I've been studying Brettanomyces for more than 3 years, because after the PhD, I went to Australia, in Adelaide, to the AWRI, Australian Wine Research Institute, to study Brettanomyces for 4 months more. So actually, I've been studied Brettanomyces for 4 years. So it's something that comes from my PhD. And then I had this idea to give wineries some tool to make their analysis by their own. Otherwise, I don't know in Greece, but in Italy is a bit complicated to have fast analysis, because you have to collect the wine samples, then you have to send to a laboratory, because in not all the Italian region there are wine laboratories. In Piedmont, for example, there are 4 or 5 laboratories, but in the south of Italy, even if there are some quite good wines, there is a lack of laboratory. So during my PhD, I understood that a tool was missing, and I had this idea to combine a deep slide technology and a culture media. So it actually only detects alive cells. On the contrary, PCR can also detect dead cells, because the Brettanomyces DNA remain on the wine.
Okay, we have another question by Dr. Dimopoulou. She asks, have you tested the VNC states, viable but not culturable, of bread? Is it possible that the self-test can't detect the cells?
Yes, actually, there is a bit of confusion on this VNBZ state, because they are really stressed. But usually our clients, they do not refer any difference with the comparative analysis in the laboratory. In the beginning, when we have developed the kit, we conducted a study comparing PCR plate counting and the self-breath, and we were able it was done on 80 wine samples, 80 different wine samples coming from different vineyards. And what we have seen that is in two cases, we had some difference with PCR, probably due to the detection limit. So I think that because the media on which Brettanomyces have to grow, because this media is Brettanomyces like this media. Some of the cells in the VNVC state can also grow on it.
Okay, and thank you very much. And another question for my part. You mentioned that the tests are strain specific.
Yes.
No,
I mentioned that
not strain specific. Sorry. Microorganism specific. But what happens with the false positives? Have you encountered a case like this?
If all of them are positive.
False positives.
False positive. No, actually, we never experienced false positive, because if something grows on it, it's impossible to have a false positive. If the only problem is if there is a environment contamination. But what we have done is to when we have developed the test, we opened the self-breath and self-micro in the winery's environment for different minutes, and we have understood that the probability to have a contamination from the environment in the very short time that you have to open the bottle is very low. So actually, we have never experienced a false positive. And one of the study case, one of the best study case, is for self wine, for a wineries that found the slide of wild yeast contaminated, but the self breath not contaminated. So they send us the self wine, and we extracted the DNA and send for sequencing to understand which species it was. So they understood that was I think I can remember if I was a pichia or something like that. So they understood which was the source of contamination, which one was probably a special equipment.
Okay, that was very clear. So we have one more question. The question is, can this product be used as a way to identify the same strain of breaths? For example, I have a small vessel of 100 liters with a desirable breath-infested wine. By taking a small inoculant of the original liquid for another, can the test identify the same strain has developed in the new wine?
No, it's not that. It cannot let you know anything about the strain type, because it's not at strain level. It's just at species level. If you want to understand if it's the same strain, you have to do, on my opinion, molecular biology analysis. And it's something that I have done during my PhD, and we are able to do it.
Okay. Thank you very much. I do not see any other question. So I don't know if Panagiotis Stamnidis wants to add anything in the conversation.
Okay, yes, but I will do it in English. in order
sorry,
also
sorry,
to...
Panayotis, sorry to interrupt. We have another question. If we have a positive wine test, we can understand from what microorganism we have contamination, wild yeast, bacteria, etc. Or
Yes,
we will have to send sample for PCR to specify the contamination.
No,
No. Can you see imagine from the self wine is made of two vials, a green one and a red one, and then there are two sides denominated. There is written A or B. If you have a contamination on B, you know that they are wild. On B of the green lid, you know that they are wild yeast. If you have a contamination on A of the red lid, you know that is acetic acid bacteria. And if you have a contamination on the B side of a red lid, they are lactic acid bacteria. You don't know which species they are. You just know that they are lactic acid or acetic acid bacteria. If you want to know the species, you have to send a sample.
So red lid, blue lid.
Red lid, green lid.
Red lid, green lid. We had red pill, blue pill in Matrix. So Panayotis, sorry for the interruption.
what I wanted to say is, for me, because my background is also biotechnology, and I have also friends of mine that were experts in real-time PCR. I have to say that it's not a very easy task to detect with a molecular technique Brettanomyces. First of all, it's very expensive, and you have to have a very well-trained personnel that knows molecular biology techniques. Just imagine that just to invest in a real-time PCR, that in reality will show you the DNA, so you don't know if for example, your bread is alive or dead. So you know that you have DNA, but you don't know if it's alive or dead. Of course, the detection limit of the method is very low with real-time PCR. But just imagine you must probably pay 20,000 euro only for the machine, and for the probes, for the molecular probes, you can have a cost of analysis of around 25 euro, 30 euro. Cost of analysis. So we're talking, just imagine that in the Greek market, it will be something around 16, 17 euros, this box. Okay, we can make some discount if someone gets many items, but it's amazing because, first of all, you don't have to invest in the other technique is flow cytometry, which is also very expensive, the machines. We're talking for 50, 60,000 euro for a flow cytometer. And then also you have to buy some fluorescent dyes. So again, they are very expensive techniques. And then, of course, it's the microbiological techniques, which you have to have a microbiology lab to do it, which is also labor intensive. At the end of the day, I don't think that a winemaker will invest 40, 50,000 euro and the personnel only to detect bread. It's from the economic point of view, for me, it has no meaning. So that's why in the most of the ones, even in the best of the best in Greece, very rarely you will see microbiological analysis. This is the reason, I think. So with this kit, we thank Simona to do this. It gives the opportunity without someone being not even an enologist in reality, to just take the box to. It's very simple, the use. I mean, it's like the self-test we do for the COVID. I mean, it's very easy to use. Simona, sorry, this is the box, and just you open it, and it has an agar slide. So you put the wine. That you put the wine till the sign that shows you here. you just close it, you shake it for 60 seconds, you pour the wine, you just leave the wine to be a little bit drained, and you close it again without touching any surface, and you leave it in a room temperature. So it's very easy, very cheap, very economic, very reliable. So I think it's a perfect solution for the wineries. And this is for bread. Sorry, this is for bread. And of course, for the self wine, there are two vials. One is for detecting cerevisiae, the one part of the slide, and wild yeast for the other. And the other vial, the one part of the slide detects lactic bacteria and the other acetic. Am I right, Simona?
The one side is for total yeast. So
Total
usually,
yeast, yes.
usually Saccharomyces grows more than the others, but
Sorry.
they can grow all of them. The
Okay,
other
okay.
sides, only not Saccharomyces can grow.
Only wild yeast. Only wild yeast.
Only wild.
Only wild yeast. Επίσης, είναι πολύ σημαντικό αυτό που είπε κάποια στιγμή. Sorry, it's also very important, something that Simona stated.
You can talk in Greek
Okay,
if you want.
Είπε κάτι πολύ σημαντικό, ότι πολλές φορές μπορεί να τσεκάρεις. Δηλαδή, λες, έχω ένα φίλτρο, μια καμπάνα, την οποία, ρε παιδί μου, τη χρησιμοποιείς κατά κόρον, την πλένεις, φίλτρο κάρτης που λέμε, τελικό φίλτρο, το τελικό σου φίλτρο πριν το φιλτράρισμα του κρασιού σου. Και θέλει να δεις μήπως αυτό τελικά έχει μολυθεί με βρετανομύκητες. Άρα θα μπορούσε αυτό να το χρησιμοποιήσεις, να γεμίσεις με νερό και να χρησιμοποιήσεις το νερό που βγαίνει από το φίλτρο μόνο και μόνο για να τεστάρεις αν το φίλτρο είναι μολυσμένο ή κάποιο άλλο μηχάνημα. Άρα μπορείς να ελέγξεις ίσως και σε ποιο σημείο γίνεται το contamination. Πολύ σημαντικό, κάτι που νομίζω κανείς δεν μπορούσε να το κάνει πριν, είναι ότι μπορείς να ελέγξεις και το σταφύλι σου, ο μούστος σου. Και έχουν ειδικό βέβαια kit για αυτό που λέγεται self-bred grape, νομίζω είναι αποκλειστικά μόνο για την πρώτη ύλη. Άρα για να δεις μήπως τελικά ξεκινάει το contamination από το σταφύλι. Εν πάση περιπτώσει, αυτό που παρουσιάστηκε εδώ πέρα είναι νομίζω κάτι, και αυτή είναι η λογική αυτών των σεμιναρίων, να παρουσιάζουμε λύσεις στον εινοποιείο, οι οποίες είναι κάτι καινούριο, είναι κάτι καινοτόμο. Αυτό παρουσιάστηκε εδώ πέρα. Ευχαριστούμε όλους όσους, εντάξει, νομίζω θα προλογίσει, θα κάνει το τέλος της τέτοιας ο κύριος Τζαχρίστας, αλλά εγώ ευχαριστώ όλους που παρακολουθούν αυτό το σεμινάριο και μην διστάσετε, τα kit, αυτή τη στιγμή η ενοανάλυση έχει διαθέσιμα kit bread, αρκετά για να καλύψουμε οποιαδήποτε ζήτηση. Self-wine αυτή τη στιγμή δεν έχουμε έτοιμο παράδοτο, από την αλήθεια, αλλά η Ιταλία δεν είναι κάτι δύσκολο. Μέσα σε μια εβδομάδα μπορεί να έχουμε σε οποιαδήποτε ζήτηση να ανταποκριθούμε. Αυτά από μέρος μου, γιατί δεν θέλω να κουράσω, γιατί ξέρω ότι όλοι μπορεί να δουλεύουν και η ώρα είναι πολύτιμη και αυτό ήθελα να πω. Ευχαριστώ πάρα πολύ και όποιος θέλει να κάνει κάποια άλλη ερώτηση, αλλιώς Αλέξανδρε νομίζω ότι εσύ θα κάνεις το τελείωμα, τον επίλογο.
Okay, so Simona, we would like from Panepo's part to deeply thank you for your contribution in today's presentations. Everything was very clear. The answers to the questions, I believe, satisfied everyone. I would like to ask how much time have been the kits available in the market? I mean, it's something new, 6 months, 1 year, 3 years, how much time?
Self-bred was launched in Italy in 2019.
Okay.
Then we had a lot of requests, but then COVID came and restaurants were stopped to buy and something else. So we were not very lucky. So actually we launched it, let's say, again in 2021 almost. I mean, it's had been on the market since 5 years. So it's quite known in Italy. We also sold it in USA, You can find it because it was an artist that was commercializing it. So if you check Self-Breath and USA, you can find an artist page. Even in Australia, so Self-Breath has been on the market since 5 years. Self Micro, Self Wine, we just launched it in the last international fair exhibition that was in November in Italy. So has been on the market since 2 months.
So good luck with that.
Thank you. I want to thank all of you for the opportunity, and I hope you enjoyed my presentation. If you have any question, please feel free to contact me. And thanks again.
Okay, so I'm going to switch to Greek now.
Yes, you can.
Okay. Λοιπόν, αγαπητοί, ευχαριστούμε για την παρουσία όλων σας αυτή την Παρασκευή. Ακούσαμε από την κυρία Καμπολόγκο ενδιαφέροντα πράγματα και από τον Παναγιώτη τον Θαμνίδη για το πώς μπορούμε να ανιχνεύσουμε διάφορους μικροοργανισμούς τόσο σε πρώιμο στάδιο στα σταφύλια όσο και σε όλα τα στάδια της νοπαραγωγής. Θεωρώ ότι όλα αυτά είναι πολύ σημαντικά στην εργαλειοθήκη του σύγχρονου εινολόγου. Συμφωνώ απόλυτα με αυτό που είπε ο Παναγιώτης, ότι πολλές φορές συχνά ελέγχουμε τα κρασιά μας για διάφορες παραμέτρους τρέχουσες, αλλά δεν είναι τόσο κοινό να ελέγχουμε αναφορικά με το μικροβιακό τους επίπεδο, με το μικροβιακό πληθυσμό σε αυτά. Άρα θεωρώ ότι πολλές φορές μπορούμε να έχουμε απαντήσεις σημαντικές και να μπορούμε να επέμβουμε πριν φτάσουμε στο στάδιο της τελικής τυποποίησης. Να θυμίσω και πάλι ότι η παρουσίαση θα ανέβει στο κανάλι της Πανέπο πάρα πολύ σύντομα. Κάντε εγγραφή για να μπορείτε να τη δείτε, να τη βλέπετε όποτε θέλετε και να μπορείτε να δείτε και όλες τις υπόλοιπες παρουσιάσεις. Θα θέλαμε να σας ευχαριστήσουμε, να ευχηθούμε και πάλι καλή χρονιά, μιας και είναι η πρώτη παρουσίαση της χρονιάς, και συντονιστείτε για τα επόμενα. Ευχαριστούμε Σιμώνα, ευχαριστούμε Παναγιώτη.
Να είστε καλά και εμείς καλή χρονιά και ό,τι καλύτερο. Καλή συνέχεια.
Καλή συνέχεια σε όλους, να είστε καλά.